Hello, folks. I want to talk to you now about skeletal muscle physiology and specifically about uh, how skeletal muscle is stimulated to contract. Okay, so we have to remember that muscles are excitable cells, and the way that any cell can be excitable is by being responsive to electrical stimuli or being responsive to hormonal stimuli, right? Skeletal muscles rely on electrical stimulation. So if we just kind of look at our, whoops, we'll look at our brain or spinal cord. So this is a little model that you can think of. This is where all of our neurons live, and motor neurons in particular are those that travel from the brain or spinal cord out to a variety of places, but skeletal muscles is one place that motor neurons go. So this is showing a motor neuron. Here's the cell body in the nucleus. Here's this long extension of the neuron. It's called an axon. We'll talk about that later. And then here it is connecting to a muscle cell in a few different places, right? Sorry, a muscle in a few different places. And I'm drawing these individual pieces as, like, think of those as, in this simple case, you can think of these as muscle cells that are being stimulated by an individual end point of this neuron. So we're going to think about first and briefly, and we'll think about it a lot more in neural tissue. How do we get a signal from here where it's initiated all the way down this neuron to the muscle cell? That's called propagation. So how do we propagate electrical signals that are initiated up here in this neuron all the way down the neuron and to the muscle cell? Propagation actually happens later too. After we get the message conveyed to this muscle cell, we actually get propagation of this same electrical stimulus all the way along all of these individual muscle cells of the whole muscle. So we'll talk about that too. Second, we've got to, once we propagate this signal from the cell body down to the endpoints here, we have to then get that signal from this neuron to this muscle cell. And that involves a process called transduction, where we actually change, in transduction, we change this electrical signal into a chemical signal so that it can traverse the extracellular space between the neuron and the muscle. And then we'll talk about excitation. So once we've done this transduction step and we now have a chemical message moving from this neuron, just outside this neuron, to this muscle, how does that chemical message then excite that muscle? So how does it pass on the stimulation of this neuron to the stimulation of this muscle? And finally, how does that excitation of this muscle cell lead finally to a contraction of this muscle cell? So there are a few different steps to think about here. Before we get into that, I just want to show you a little schematic that looks something like this so we can draw out how these cells remain responsive or maintain their ability to respond to electrical stimuli and to respond to any stimulus, actually, but how that can then be converted to an electrical stimulus that travels all the way along this neuron. So I'm going to go back to our sketchbook here, and we'll look at that in a little more detail. Hopefully this looks similar to what I just was showing you. I want to just show you that inside muscle cells and inside neurons and inside most cells of your body, they are maintaining some homeostasis of a variety of things, but one really important thing is charge or electrical potential, right? So all cells maintain an kind of asynchronous distribution of charged ions across their cell membrane, so that in general, the inside of a cell, here we go, inside of a cell is relatively electronegative, okay, all over inside of most cells. It's relatively electronegative in here. Same with this. Inside of this muscle cell, it's relatively electronegative. And out here, it's relatively electropositive, okay, out here. That simple difference in charge right there is, as you know, potential energy, right? It's the energy of position. So this separation of charge represents potential energy. Hopefully you remember from Biology 160 that the way that is maintained largely across this, and the best known, the way that's been most studied, the way that this is maintained across this plasma membrane is via the activity of pumps, right? So we'll just blow up that region of cell here, and we're seeing some plasma membrane, and in that plasma membrane are some pumps. And I'm just going to draw a pump. Remember, this is any kind of thing that's called a pump, right? Is a protein, and it's using ATP to power it, right? So what these pumps are doing is actually exchanging a couple of ions across their membrane with the help of ATP. This sodium potassium pump, if this is a sodium potassium pump, is taking in potassium, and as it does that, it's ejecting sodium. And for every 2 potassium that come in, 3 sodium leave. And it's doing that all the time. It's creating an asymmetry in these charges, an imbalance in these charges. And then all cells also have a lot of what are called leak channels. And the most common leak channel, sodium leak channels are quite uncommon and not very many of them, but there are tons of potassium leak channels. Notice I'm drawing this channel as just this open thing here. There's no lid on it. There's no regulatory mechanism on it. It's just open, right? So this is a leak channel. So potassium, that means, can leak anywhere into or out of the cell based on its concentration gradient. So the upshot of all this is that in general, there's a lot of sodium outside the cell and held against its concentration gradient out here. And there's not very much inside the cell, right? In general, the potassium concentration gradient is not large. Now there is in general a lot more potassium inside the cell than there is outside the cell, but the electrical potential, or sorry, the driving force on this potassium is not very strongly outward. It is a bit, but not very strongly outward, while the driving force on sodium is very strongly inward. And then there's one we haven't mentioned yet. There's a lot of calcium outside of cells and very little in the cytoplasm of a cell. We'll see, of course, that within muscle cells, we talked about how the terminal cisternae, which is the fused SER, holds a lot of calcium within it, but that's not in the cytoplasm. That's not free floating in the cytoplasm. That's contained within this storage vessel, right? So in general, lots of calcium outside and very little calcium inside, and no leak channels here. So basically the driving force on sodium is very strongly in, and on calcium is very strongly in, and weakly, very weakly out on potassium. But this is how cells maintain an electrochemical gradient. They force these charges against their concentration gradients, and they maintain the separation, especially of sodium, so that on the whole, it's relatively electronegative inside a cell and relatively electropositive outside the cell. Okay, that's going to kind of set us up for looking at the rest of this this lecture, or at least the next little bit of this lecture. And we're going to see here just kind of a blow up, a large version of what I just did, right? Showing this electrochemical gradient. And what you can see here is all along, here's one thing I did draw, the potassium leak channels, but these cells are also filthy with semi-permeable channels. What that means is at rest, these channels are closed. You can see a sodium channel here, and a potassium channel here, and at rest, these are closed, right? Now, there are a number of different, well, there are only three really, different stimuli that can cause channels like these to open. The one we're going to be talking about, at least when we think about the propagation of a signal along a muscle cell, or sorry, along a neuron or along a muscle cell, are those that are affected by voltage change, right? Or by the absolute voltage or electrical potential that they're experiencing, right? So that's what really governs the behavior of these, what we call gated channels, these semi-permeable gated channels. These channels can open and they can close based on changes in the voltage that this channel experiences, right? So just right outside and right inside of it. Okay, so this is now showing plasma membrane here, these blue cells, blue phospholipid molecules, and just tons of gated channels in here, right? And it's showing you the outside and inside this this charge separation, right? So it's a bit like a battery or a capacitor in that way. Right? So the way that these cells, that all cells communicate with each other, at least in this neuromuscular communication, is by altering the potential of each other. So here again is like a different view of those electrochemical gradients, where you can see there's a lot of potassium outside, inside, and not much outside, right? And there's a lot, and so we have this like large chemical gradient this way, but and a large potassium, a modest potassium gradient this way, but that's kind of buffered by a potassium electrochemical gradient, right? So it kind of suppresses this this outward gradient. So there's some outward gradient, but I would say the arrow is kind of not doing it justice. It's really not even that strongly outward. And this is showing you at negative 70 millivolts, which is about the resting potential of most cells. That's about the electrical difference from outside to in that most cells are operating at on average. This is showing you sodium, where we have a strong inward pull and a strong both electrical and chemical, making a very strong electrochemical gradient for sodium inward, right? Okay. Now you can see it doesn't for sodium or for potassium. Approximately although there is a push on potassium outward, look at the difference between these numbers, negative 70 millivolts to negative 90 millivolts, a difference of only 20 millivolts. And we would basically be at equilibrium for potassium, where we would get equal movement of potassium in both directions. So not a very strong, as I said, not a very strong push outward really, right? Which is really, they just goofed on these arrows. What the heck, man? This should be really small and this should be enormous, because look at this difference here, right? Negative 70 millivolts and positive 66 millivolts, right? I mean, that's what is that, 1300 or almost 1400 millivolt difference. 130 to 140 millivolt difference, right? You have to get that. That's how strongly that difference is before you would get approximately equal movement of sodium into the cell and out of the cell, not till you get to plus 66 millivolts. So really the driving force here is quite strong, right, on sodium inward. And we'll look later, but we'll see that in terms of calcium, it's even more strongly inward than it is for sodium. And that'll be important. That'll come up later and be important. Okay, so what do muscles do to produce a contraction? Well, I keep talking about how a neuron has to send a signal to a muscle cell, has to propagate a signal to a muscle cell, or propagate a message to a muscle cell. That message is called an action potential. And we're going to talk a lot more about action potentials, because it's the way that all neurons communicate with any cell that they come in contact with. And the way they communicate with each other is they rely on action potentials to bring to move a message along themselves. Then we also see that there's basically an action potential that spreads across a muscle cell once this signal from the neuron has been transduced into a signal to the muscle cell. And so that's happening via those transverse tubules in the excitation phase. And then once that happens, the thick and thin filaments, the actin and myosin filaments, interact and produce a contraction. So what does that look like? We got to think about the roles of sodium and potassium and calcium in this. We'll talk later about where does the energy to contract come from. Right now, I want to think more about this action potential. And we just define this as a change in the transmembrane potential of an excitable cell that's propagated along that cell. So it moves from one place on a cell to another. In terms of action potentials, they always move from their point of origin to the end of a cell, and there's some directionality. Cells can control the directionality of this basically just by the behavior of their gated channels. Right? And this action potential is a message that tells the cell to contract. Right? We can initiate this by an action potential by changing the membrane's permeability to sodium ions, and a number of ways that can happen, right? If you're thinking about a neuron, it could be a mechanical stimulus, it could be a chemical stimulus that changes the permeability to sodium, or it could be an electrical stimulus that changes the permeability to sodium. But however that happens, the way that permeability to sodium is changed, that means that channels are opened, right? That is how we change the permeability of a cell. Channels either have to open or close. That results in a change in permeability. And so when a cell is stimulated, gated sodium channels open, and they become very permeable, much more permeable to sodium when they're open, of course, than when they're closed. And that allows sodium to rush down its concentration gradient. And if you remember, the concentration gradient is very strongly pulling sodium in. So sodium is very highly concentrated outside the cell, and both the electrical and the chemical gradient are for sodium to move into the cell. So once a cell is excited and those gated channels do open, we get an influx of sodium ions into the cell. So here's what that looks like, right? Here's a cell at rest. We have potassium moving in and out through the leak channel. Now some stimulus arrives and causes these gated channels to open. When we think about a stimulus arriving at a neuron or something stimulating a neuron, tracing the origin of that stimulus is sometimes tricky, but it almost always is due to binding to some chemical, right? That causes these, it's almost always due to binding to some chemical that causes these sodium channels to open, right? At least in the receptive field of the neuron, it's usually the binding of a chemical. But then all across the rest of the neuron, once you get away from its receptive field where it's actually picking up or receiving a signal, once you get away from that, these are now no longer stimulated by chemicals, but they're stimulated by voltage change, right? So these that you're seeing here that they're drawing for you are somewhere outside the field of a receptive field of a neuron, and they're opening in response to a change in voltage, okay? I just want to jump back here real quick and outline, like, what is a receptive field? What is that? Okay, so if we're back here at our cell, this is where the receptive field of a neuron might be, right? So maybe I'll just say, well, let's see, do yellow. So this might be where a neuron, somewhere along, all the way along here, is often where neurons are like receiving signals, right? Or receiving stimuli. And along here, so this is like the often, and this is, you'll see this is very diagrammatic, and when we get to the nervous system, you'll say, oh, that was very simple-minded drawing. Yes, but that's the receptive field. So that's where you often have gated channels that respond to chemicals. And then all along the rest of this, all along here is where we have gated channels that are responding to voltage. all the way to the end of this thing, right? This is like where you have voltage gated channels. So we'll see this, that the same thing more or less is true of muscle cells, in that, where should we go here? This, right along here, is the receptive field of the muscle cell. This is where you would have gated channels responding to a chemical signal. And then all along the rest of this muscle cell, yeah, all along the rest of this muscle cell, right? I'm just going to draw out here. This is where you would have gated channels that are responsive to changes in potential, right? Or electrical, that are responsive to electrical stimuli. Okay. Let's jump back now to our lecture, and we'll see when these voltage gated channels open, sodium rushes in, and that causes a large depolarization of the cell, right? When sodium rushes in. And I'm going to show you this in a minute. Now, potassium channels respond to the same stimulus that sodium channels do. So they respond to a change in voltage, and these are called low voltage gated channels. So they respond to depolarizations away from their resting state. They respond really early on to those changes away from resting. And the difference is they simply open quite slowly. So whatever the mechanics of these channels are, or however this channel is constructed, the result is that while these sodium channels more or less spring open, these potassium channels here are kind of just leisurely opening like this, right? So we actually get a really strong influx of sodium and a depolarization of the cell before we get the opening of potassium channels. And once that happens, we get potassium rushing out of the cell. Right? Okay, so let me just quickly show you that. Here we go. So here we're looking at an action potential. Here we're looking at the voltage in millivolts that we can measure across the membrane of a cell. Here we have -70. That's basically the resting potential on average for most cells. And you can see how this goes up more or less. It changes in a nonlinear fashion, more or less, but whatever, right? We go to 0 and then to the plus, and then here's plus 40 millivolts, right? Here you can see that time, elapsed time, and this is in milliseconds. So you can see that this signal, this action potential that propagates all along a cell, passes a given region of membrane like that, right? Lightning fast, milliseconds here. This whole depolarization away from resting and then getting back to resting, that whole thing takes 4 milliseconds on average, right? Really fast. And now this is showing you, here we are, right? Here we are resting, this cell is resting. Now, here's this arrow, stimulus. So something is stimulating a cell, causing gated sodium channels to open. Now, if enough sodium channels open and enough sodium rushes in, because remember, sodium is a positively charged ion held at very high concentration outside the cell, and it's at low concentration inside the cell. So when you make a cell permeable to sodium, it's going to rush into the cell along its concentration gradient. That means it's adding positive charge to the inside of the cell. So if you're adding positive charge, you're going to be going up. You're going to be depolarizing, going towards 0, because you're way down here at -70. So we have this sodium rushing into the cell, adding positive charge. If that happens quickly enough, and enough of it comes in to get to, like, what is this, -55 millivolts. Remember I said that these channels are low voltage gated channels. At about -55 millivolts, that's when all the voltage gated sodium channels pop open fast. And when all the potassium, voltage gated potassium channels start to do their leisurely opening, right? So if we get some chemical or mechanical stimuli opening enough channels in the receptive field of a neuron to cause to get the voltage of that cell, the transmembrane potential, to this threshold value of 55, then all of the neighboring voltage gated sodium channels open and we get this huge influx of sodium. And what you're seeing in this depolarization is this is because there's a huge influx of sodium going all the way up to this point here where we'll talk about channels going through a variety of states, but basically they're closing here. And this is about when potassium channels are finally getting themselves open. And so now we're at plus 40 millivolts, which is way off from the equilibrium potential of potassium, which we saw was like negative 90, right? And so now potassium is going to rush down its concentration gradient. So because there's so much potassium inside the cell and very little out, we're going to get this huge efflux outflow of positively charged potassium ions. So here, to get a depolarization, we had an influx of positively charged ions. So it makes sense that an outflux, an efflux of positively charged ions is then going to allow us to repolarize and go back to our resting state. But in fact, because these potassium ions or channels are so slow to open and close, we get this refractory period where it dips down here to like negative 90 or close to negative 90 to its equilibrium potential before the sodium potassium pumps bring it back up to its resting state here. Right? Okay, so that's a bit of complex. You may want to watch that a couple times and think about it in terms of what's going on with these cells, right? Because we want to understand kind of how this whole process is happening from neuron to muscle, and then we get to muscle contraction here. Okay. So maybe this is a good place to stop and you can kind of review that, and I will just pop back out here to our sketchbook, and right, you can see that just remind yourself, receptive field, that's where we have the gated channels that are opening in response probably to a chemical in this case, and or the binding of some kind of chemical to them. And then all along here in the orange, this is where we have the voltage gated channels, those that are responding to a change in the voltage. And specifically, now we know a change in the voltage that gets to threshold, or in these cases, about negative 55 millivolts. And at about negative 55 millivolts for both the sodium and potassium channels on this neuron, as well as those all along the sarcolemma, they're going to open in response to that. Okay. All right. More on this in a future video. Please let me know if you have any questions about this.